I'm here to talk about biomarkers in clinical development, enabling drug discovery. And also I have some thoughts on personalized medicine. But first, I do have a few disclosures, and I'll do a little bit of an introduction. So my name's Jeff Dage, as Courtney mentioned. I'm at the Indiana University School of Medicine, Department of Neurology. I've been there only about 2 years. Before that, I was at Eli Lilly and Company, which is why I have some Eli Lilly and Company stock, and I still do some work with them as far as public speaking. So, and I spent about 20 years in doing drug discovery at Lilly, and before that, another 8 years at Aventis Pharmaceuticals. Most recently at Lilly, I was in charge of developing biomarkers, biomarker strategies, and assays to implement for the neuroscience clinical development programs, where I discovered and developed the phosphorylated tau assay that people are using out of blood. And so the blood-based biomarkers for Alzheimer's disease came out of my lab at Eli Lilly, and we've been using it pretty extensively in tailoring as well as now treatment response, which is pretty exciting to see. Hopefully you had a chance to read all of my other disclosures. I'll move right on into what a biomarker is. And I put this in, it's really kind of simple. Biomarker is pretty much anything you can measure. And I point this out because a lot of people use the term biomarker, and it's very generic. It can mean just about anything. The way I use it, I'm usually talking about biochemical biomarkers out of biofluids, and that can be CSF, plasma, serum, urine, but I will do other things out of tissues, skin punch biopsies, and things like that. But it really is a generic term, and it can be anything, simple measurements from body weight or blood pressure, cholesterol, which we all have measured often, maybe, or heart safety monitoring, so cardiac troponin I for myocardial infarction, and I mentioned already phosphorylated tau, which we developed at Lilly for Alzheimer's disease. But it really is just about anything you can measure. And so the way I think about this, and I thought I would really stay focused on drug discovery, drug development, and clinical trials that pertain to investigational therapies. And so there's some general categories of biomarkers, and so now you become a little bit more specific. In early clinical development, it's really, really important to have biomarkers of target engagement and or pharmacodynamics. And the target engagement is really a direct measure that your investigational therapy is actually binding to the target. And so it's very direct. Anything else is really pharmacodynamic. And so when you measure the product of an enzyme, like you might in a BACE inhibitor program, that's a pharmacodynamic measure. When we measure a lot of A-beta species in our C-negacistat trials or our BACE inhibitor trials, those are pharmacodynamic measures. Still very, very useful, but in the closer they are to the target, the better they are at helping you to identify and set doses in early clinical development. Without that, you don't really know if you've gotten to the target and if your molecule is doing what you think it's supposed to be doing in people, regardless of how much you've tested it in other places and maybe even other diseases. You really need to understand these or have these measures to do a good job of setting dose. Then there's pathophysiological biomarkers. I mentioned phosphorylated tau, so that's a great example of this. It's a measure of a physiological system or process that's directly related to the disease process. And so when we worked with phosphorylated tau measures, we were correlating it and using it to predict amyloid PET, which was an FDA-approved test for amyloid positivity in Alzheimer's disease, as well as tau PET, which is an FDA-approved diagnostic to determine tau pathology in people being evaluated for Alzheimer's disease. And so it was a measure specifically designed around the disease process itself. Now, what can you do with those pathophysiological markers is you can use them to identify patients. And so if you're going to target things like in Alzheimer's disease, an amyloid targeting agent or a tau targeting agent, you'll want to know that there's evidence of that the pathology is present in the patients you're going to treat. And so that's where you would use a pathophysiological biomarker in patient identification. And that was initially done in a lot of Alzheimer's disease clinical trials. Early on, we would enroll people who were mild or moderate symptomatics, And then we started using amyloid PET, and we could do amyloid PET positive symptomatics. Now we're enrolling phosphorylated tau positive subjects into clinical trials, even asymptomatic subjects. And so now you can move earlier and earlier in the disease process because you can identify a pathophysiological form of the disease in these biomarkers. And then you can move on to clinically predictive, and that's where a pharmacodynamic measure in a treatment trial is associated with or correlates with a clinical endpoint. And so it predicts a clinical benefit. It can also predict tolerability or safety. And as you develop this, it can become very, very important, such that you can then totally redesign the way you do clinical trials, because oftentimes you're powering for a highly variable clinical endpoint, which can require a long time. In the case of Alzheimer's disease, it was 18 months and thousands of patients. Now we can do something on the order of 20 patients to look at pathophysiological changes, which is pretty exciting. And so you can kind of see that there's a huge difference in how rapidly you can do clinical development. And so then it becomes, if it is clinically predictive and you can prove it across several molecules, it can become a clinical endpoint. And that's an association with how the drug is actually making the patient feel or function. And this is really up to convincing regulators that it's a clinically meaningful endpoint. But that's really, the ultimate goal of some of these biomarkers is to try and get to that point, because it really helps enable clinical development. So now I wanted to highlight an example or a case study. And so this came from a small biotech company, Bials, in France. And it's titled The Clinical Disaster. And it was back in 2016 where they were doing a phase 1 study of FAH, fatty acid anandamide inhibitor, hydrolase. It's involved in the synthesis of anandamide. And so anyways, they were in this phase 1 study, and they had administered the drug in a dose escalation, and they had a person die. And they had several other people get very, very sick. There was a lot of criticism, a lot of investigations. They had absolutely no target engagement or pharmacodynamic measures in the study, because phase 1 safety studies typically are all about safety and pharmacokinetics. So safety is the fundamental goal or the primary outcome of some of these phase 1 drug studies, and pharmacokinetics is typically a secondary outcome. So those are kind of what's required, but not having target engagement and pharmacodynamics can lead you to overdosing or dosing beyond what is maximally impacting the target, which can lead to off-target effects. And so we'll come back to this case study, but it caused a huge problem because there was a lot of FAH inhibitors in clinical development at the time at multiple companies, including Johnson & Johnson and Pfizer. And when this happened, the FDA put everything on hold. The whole class was put on hold. Everybody had to stop dosing. They had to do a lot of investigations into what happened, why did it happen, is it compound related, is it target related, what's the overall impact to patients. And so this was a huge issue, not just for this one company, which experienced a death in their clinical trial, which is horrible, but it impacted the entire industry of people investigating these drugs. So sometimes another trial can impact your trial in totally unexpected ways. And I think it's really important to think about that and think about the questions they were asking about. Why didn't you include a pharmacodynamic measure so you knew the impact you were having on the target? That was one of the main questions people were asking. And so one thing I want to focus on is this back translation concept. And it kind of fits with what I was just talking about with this experience that somebody had in the clinic. What does it mean? Going back from our clinical findings, going back to discovery, back to in vitro and in preclinical models to understand, can we associate what we saw in human clinical studies with take that learning back to the preclinical and advance our knowledge in order to kind of iterate. And in the context of biomarker development, that's kind of what happens. In this case, they didn't have a biomarker. So you come back and you look at this, well, what should we have measured and how could we have measured it? And you can develop a whole strategy around this. This is kind of what I was doing a lot at Lilly. You start with a strategy and then you develop a plan and then you design assays to measure very specific things. And then you validate those and put those into your clinical protocols. And really, that's really designed to help you establish doses in phase 1, in phase 1B, and then have confidence that in phase 2 or your proof of concept study, regardless of the outcome, regardless of the outcome, if it's a positive study, that's great. You know how hard to hit it and what to dose. If it's a negative outcome, you tested your hypothesis and you can have confidence in the result. And so there's only upside to having these kinds of measures. And this back translation happens all the time. When you think about the whole story of Alzheimer's disease and amyloid targeting therapies, you can see the evolution of this over time. And so what happened with some of these other FAH inhibitor programs? I mentioned several were put on hold. Well, they did a lot of investigations and they decided that it was probably related to that compound specifically and some off-target effects. And so they released the clinical hold, but then who's going to go next after some company saw somebody die in one of their clinical trials? Who's going to go and target this again and take that risk? In this case, it was Johnson & Johnson. And so they came much more prepared. So they did measure the concentration, the PK, had a very sensitive method, but they also measured a whole bunch of biomarkers. They measured FAH activity in leukocytes, so in the periphery, as well as concentrations of several anandamide metabolites in plasma. And then they also measured some of these arachidonic acid and some of these other metabolites were also measured in CSF. So the drug target itself was in the brain. They measured a whole bunch of peripheral biomarkers, a whole bunch of CSF biomarkers, and then they even developed a PET imaging agent specific for this enzyme. So it's enzyme occupancy. So this is displacing the active site with the drug. So the PET tracer binds the active site, it gets displaced by the drug, and you can see occupancy. And so what you think about in this case is direct target engagement. That's what this PET study was showing. And so they had the PET study, they had a whole bunch of target engagement biomarkers, and they had pharmacokinetics. And you can look at this data and see very clearly on the left side's the PK, in the middle is the biochemical measures or the biochemical biomarkers for the whole pathway, substrates, products, as well as some side products. And then you'll also have direct target engagement measured by PET. So they spared no expense when they went back into the clinic because they wanted to be very confident that they can identify the dose. And what they saw here is a very nice correlation between what they could measure in the CNS and the peripheral measures. And that's really important from a biomarker perspective because it allows you then to use peripheral sources in order to understand your pharmacodynamics and avoid doing lumbar punctures in everybody. And that's important for phase 2 studies, especially when they go much larger and multi-site. And so here you also see that at almost all the doses tested, you had really strong enzyme occupancy. So at pretty much every dose, they were hitting above 80% occupancy. So they were hitting the target really, really hard. And you can see at the 10 milligram dose, they're close to 100% occupancy. And so it shows down here in the table, the trough levels were in the 80s. So they went ahead and they took this 10 milligrams into several actually studies, and they dosed 10 milligrams, showed that it was declining to 80–85% at trough. And here they did several studies. This is just a social anxiety disorder study, and what you can see in the left-hand plot is the anandamide metabolite in the trough concentration. So a very beautiful PK/PD response. Where you can associate drug levels with pharmacodynamic action. But unfortunately, you see on the top right, there's very little efficacy. You don't get any separation between treated and untreated. And so they didn't see any efficacy, so the study actually failed. In a post hoc analysis, so this is my counsel on trouble, right? After you do a big study like this, you want to maximize your benefit. You do all kinds of analyses. post hoc, and you convince yourself maybe at this one stage, I can identify some people that showed an improvement. And that's what that bottom right bar chart is showing, that they had evidence of a moderate benefit in the cases of some people who responded with greater than 30% improvement. And so then they thought, well, maybe we can redesign our next study. And their plan was to explore higher doses of this therapeutic agent in order to try and see that effect. And so I put a question on there, what would you do? And I hope your answer is you wouldn't do higher doses. Once you're dosing at 80 to 90% occupancy, there's really no reason to go higher. There's only going to be more side effects. I don't think they ever did, by the way. Anything that used these compounds in the future always went a little bit lower. But that's a little case study just to kind of show how trials by other people can impact what you do and the extent to which you do it. And it's something to take into account. And so I'm going to skip through this Alzheimer's disease. I'm going to talk a little bit more about it because I wanted to talk about biomarkers in Alzheimer's disease. It's really changed the whole landscape of how Alzheimer's disease has been diagnosed and now treated over the course of the last 25, 30 years, maybe even a little bit more. And that has to do with the PET imaging, which I mentioned, all the way through CSF measures, which now are FDA approved just this last year to aid in the diagnosis of Alzheimer's disease. And in the upper right-hand corner is data from my own lab where we discovered this phosphorylated tau in blood test to identify high Alzheimer's disease neuropathologic change. And now you can do this all in blood. And so you can see the migration from very complicated, difficult biomarkers to get, to come up with, but also then to do lumbar punctures, pretty invasive, all the way through now a simple blood test. And it continues to be refined. And how it's been used in clinical trials, this is data from Lilly, where I was part of this publication on their recent donanemab study. This was the phase 2 study. And you can see very clearly that with donanemab treatment, you see a pretty nice response within 12 weeks of treatment, which is pretty impressive. So it's a 1.5-year study. You can see a pretty rapid response where phosphorylated tau is going down. Now, interestingly, this is an amyloid targeting therapy. So you're removing amyloid plaques, yet we're seeing downstream pharmacodynamic action in the phosphorylated tau levels. And that raises a whole bunch of questions on the interpretation and a whole bunch of finer details. I also am showing on the right-hand panel another biomarker that we measured, which was GFAP, or glial fibrillary acid protein. And that's a marker of activated astrocytes. And interestingly, when you remove amyloid, you also see reductions in this astrocytic factor, which is also really interesting to think about the impact downstream markers of disease and the consequences of amyloid plaques in the brain. So it's really exciting data, and it's really exciting to see pharmacodynamic responses to these amyloid-removing agents in clinical trials. We did measure other things, and this is my reminder to have you always say and report the things that didn't change and the things that didn't work, and because it's just as valuable to learn from these things that don't show an effect. And so on the left, we expected to see a decrease in neurofilament, but we didn't. They both are continuing to go up as we expected for the placebo. And A-beta 4240, we would expect to see an increase in the 4240 levels because it goes down with disease. But our assay here was had too much variation to detect the change because the effect size for A-beta 4240 is only 10%. And so it's really small. And so you need a high precision assay. And so in this study, our assay was insufficiently designed to measure the pharmacodynamic response in this test or in this study. So that's something to keep in mind. The assay design matters, and you should really keep in mind the effect size you expect. I'll go through this even faster because this is ESI, but they showed the exact same thing. And so anytime you're doing biomarker results and you're seeing pharmacodynamic action, you'll always want to see it in multiple studies or multiple compounds. That gives you a lot of confidence in the findings. And here they're actually seeing increases in 4240 because they measured it using a different assay, a mass spectrometry assay that was much higher precision. And so they can detect the changes that we expected to see but didn't. So that's really highlights the importance of assay design. And then the phosphorylated tau on the right is also showing a very similar response like we saw with our phospho-217 assay. And that's it. That's a whirlwind tour of biochemical biomarkers. I tried to go fast so we had at least some time for questions before Tatiana, I think, shares her part. I'm going to look at the questions and answers, see if I see any. Somebody please tell me they heard me talk.
Yes, we could hear you talk.
Okay.
You can also raise your hand if you would like to ask your question out loud rather than typing it.
Yeah, I went really fast, so if you have questions, please do ask. I can keep rambling for a couple of minutes. There's the use of these.
Yeah,
the use of these tests are important. Oh, here's a question.
It's me, Jeff. It's Lori.
Oh, hey, Dr. Goodman, how are you?
Good. So I had a question of just a general question about advice to who should you partner with to try to develop your biomarkers? I mean, what's a good team to partner with?
Yeah, I think that's really hard. So if you're talking about interventional trials that are not therapeutically minded, then identifying really good labs that can measure the disease-related biomarkers that are most widely used for your disease area. And so that's usually some academic lab. In the case of Alzheimer's disease, a lot of people use the University of Gothenburg, Sweden, or they go for the University of Pennsylvania and the Alzheimer's disease neuroimaging study, Les Shaw, and for all, especially for Alzheimer's disease, they use NICRAD, my own lab at Indiana University. But you can identify that based on the disease area. If you're going for a therapeutic trial and you're going into a first human dose situation and you want target engagement and pharmacodynamic measures, don't wait until you're writing the protocol. All the work that I did at Lilly, it took me about 2 years to develop the plans and the assays. And so you need to partner with somebody at the very beginning to kind of think through how you're going to do this. What are you going to measure? Sometimes you can find off-the-shelf assays, but you have to validate them. Don't go into a trial with assuming you're just going to buy a commercial kit and use it. Most of them don't measure what they claim they measure. So you absolutely, if you're going to do clinical research, you have to validate your assays.
Great. That's really helpful. Thanks.
Oh, here's a question. What is your caution for someone, particularly at the junior investigator level, who is proposing to test a method or technique as a potential biomarker? That's a great question. I think when you're going after testing a biomarker, focus and be very careful about the context of use. We did a whole bunch of, and then we have a whole work group around this in the biofluid biomarker professional interest area of the Alzheimer's Association. But be very specific on the context of use so that you can design the trial specifically to test and support that context. You can do that not in a prospective trial. You can do it using stored samples. So Dr. Foroud will talk about the biobanking. When you do a trial, store the samples because you might find out later you want to use those. And so you can do this biomarker investigations and qualifications on stored samples. The FDA accepts that and will allow that for supporting evidence. And so focus on the context of use, design very specific studies to support your context of use, and take advantage of the biobanks that are around the world to do that. So you don't have to go to patients and take a lot longer and more expensive. I think maybe I have time for one more.
Dr. Conlett put in the chat. Can you tell us a bit more about ADNI?
I
Oh,
was
yeah.
asking about the Alzheimer Disease Neuroimaging Initiative. I know you're currently at a conference, and maybe you can fill us in a little bit about what it is and partnering.
Yeah, so in this case, I'm at the Dominantly Inherited Alzheimer's Disease Network, or DIAN study. It's for the autosomal dominant forms of Alzheimer's disease. It's a study run out of Washington University in St. Louis, or WashU, and they have a clinical trial unit. And so we're here talking about, it's a target validation symposium, and we're talking about what are the right drugs to go into this population, what's the right time to treat, what kind of biomarkers do they really need if they're going after some of these different therapeutics. And so this symposium's just, it's basically 2 days of, there must be a couple hundred people all thinking about the same thing and how to design these kinds of studies in a very specific population of autosomal dominant people who we know approximately when they're going to develop symptoms, so we can think about how to treat early. In the case of the ADNI study, it's the Alzheimer's Disease Neuroimaging Initiative. That study's been going on for over a decade, really designed to develop these imaging biomarkers in Alzheimer's disease. And now it's been taken over by the biofluid biomarkers, which I'm excited about. And so now the whole thing is they're doing a rewriting or getting a renewal, hopefully. And the renewal is going to be very heavily laden with biomarkers in the biofluid space, and especially in plasma, as well as digital biomarkers, which I didn't talk about, which are ways to measure physical function and things like that in people. You think about the smartwatch you have and the accelerometer or the heart rate it measures. Same with your cell phone or your smartphone. There's also voice. You can actually talk in your phone, tell a little story, and it can pick up subtle changes in your nerves and your vocal cords to pick up cognitive dysfunction early, or neuronal dysfunction early. So there's a lot of really exciting, what they call biomarkers. I don't know if those are biomarkers or not, but certainly people are working on them, and it's super exciting area. I think with that, I'm out of time.
Wonderful.
So I'm going to turn it over to Dr. Foroud.
Thanks, Jeff. All right, you better go continue to solve the problems of early onset Alzheimer's disease. So I am also delighted to be here. Can you all hear me?
Yes.
Okay, perfect, because I know earlier there was an echo. All right, thanks so much. All right, so let me do the exercise of sharing my slides. Can you see my slides? Do you see it in the
Yep.
In the right display? Perfect.
Yep.
Okay. So I'm going to do the same thing as Jeff. I'm going to use my first slide to say a little bit about myself, and I'm going to use the second slide when I talk about disclosures to tell you a little bit about the research that I do. And then I'll maybe take us down a little bit to a lower level and hopefully get a bit more discussion around the mechanics of how to make sure you have the materials to do the kinds of studies that Jeff was describing. So my name is Tatiana Foroud. I am a geneticist. I'm in the Department of Medical Molecular Genetics at Indiana University School of Medicine. And unlike Jeff, I've had a far less interesting transition across a number of institutions. I've been here for all of my professional career. So here as a faculty member since 1994. So let's make sure. So in the disclosures, it kind of gave me an opportunity to tell you a little bit about what I do, and perhaps give you an opportunity to sort of put in context as I tell you a little bit more about samples and thinking about future studies. So what I do is a lot of work around biorepositories. And so my disclosures here and how I'm funded hopefully helps provide some of that context. So I work with fluid biomarkers and the samples that are going to be needed for those. And so my funding comes heavily from the National Institutes of Health. And that's because I have a couple repositories supported by them, one in the area of Alzheimer's disease by NIA called NICRAC, and another from NINDS called Biosend, and also partner with the Michael J. Fox Foundation around Parkinson's disease, and the Parkinson's Disease Foundation again around Parkinson's disease, as well as Cohen Veteran Biosciences. The other variety of areas that support the research that I do, concussion for the NCAA and DOD. And then as you can see, when you have principles around biospecimens, sometimes you end up working in other areas. So also do a bit of work around the Children's Tumor Foundation. So part of what I'll really focus on is the mechanics. How do you make sure that you're collecting what you need in order to be able to propose the studies that you want to be able to do, and how do you create maximal flexibility. So Jeff's provided the meat of what you can do once you have samples, and I'm trying to give you the meat of how to make sure you've got what you need to do those studies. Okay, so I too want to put us a little bit in the context of where are you in that pipeline and where's the work that you're doing. So you can work all the way from discovery all the way to qualification, and Jeff's given us a great whirlwind as we think our way through about a number of the areas when we start thinking about effective treatments and some of the pitfalls. I'm imagining that many of you are potentially collecting samples that are going to be used in the discovery phase, or potentially could be. So that's around the idea of having standardized sample sources, bioinformatics in terms of being able to analyze data from those samples, identification, pilot validation, and detection methods. So I'm going to be perhaps more in that early part in what I'll be discussing. So I thought I'd take a moment and hopefully having seen some of the work that Jeff described, I thought I'd say, let's take a deeper dive into fluid biomarkers. How do we collect the right samples, and what else might we want to study? So that's the direction in which I'm going to take our discussion, and I'll make sure that I'm too leaving us. I've got 2 points of questions that we can do within my talk. So again, taking a step back and thinking about blood-based biospecimens, we've heard quite a bit from Jeff around plasma. Just wanted to remind all of you, there are other things you might want to be collecting. And as a geneticist, I'll talk a little bit more about what DNA might be able to do for you. But just to remind you, if you're thinking about DNA, and you're thinking about the kinds of studies you can do, there are multiple sources that you could use to get DNA. A lot of what we think about is blood, but just want to remind you that sometimes other tissues can be very important depending on what you might want to study. So for example, you can obtain DNA from brain tissue. And if what you're interested in, for example, is methylation, it might be very important for you to be able to study a primary tissue where you're looking for changes. Saliva can be a really accessible source for a DNA sample. You might get a more limited amount of sample, but if collecting blood is something that's more difficult, just wanted to highlight that there are other ways that we can obtain a sample for DNA. And there's a wide variety of things that you can do with DNA. Obviously, genotyping, I'll talk a little bit about ApoE later. Things that you can do like genome-wide association studies or SNPs, sequencing, whole exome and whole genome, and a lot of interest around epigenetics. So just sort of giving us a flavor that there's a wide variety of things we can do. And sometimes where you get the sample, blood, tissue, saliva, can make a difference of what you'll be able to ask later on. Similarly for RNA, you can obtain RNA from blood if you're looking about peripheral samples, but often what you might want again is RNA from a primary tissue where you're expecting and anticipating that you might see differences in expression. So often in neurological disorders, we're thinking about brain tissue, although it doesn't have to be exclusively. So again, what you can do with that RNA could be things like total RNA and microRNA. People think about transcriptomics. A lot of interest more recently around single cell work. So not trying to do bulk RNA from a tissue, but just perhaps we want to look at particular cells. This has been incredibly powerful in the space of cancer, but can also be incredibly powerful when we think about brain tissue and when we think about regions and again, specific cell types. There's really tremendous opportunities and work being done in that single cell space. You've heard a lot in the context around plasma. So Jeff described to us quite a bit around what you can do with plasma samples. And I'm just sort of reinforcing it's been incredibly powerful in the space of Alzheimer's disease, whether it's looking at A-betas, taus, neurofilament. A lot of work's been done around blood-based biomarkers, again, because of their accessibility and the ease with which that can be collected. I'd just like to remind everyone, we spend a lot of time talking about plasma, but serum can also be very valuable. A number of basic chemistry labs would specifically use serum. So, and we sometimes don't know when developing an assay if it's going to be better to optimize it in serum or better to optimize it in plasma. So I like to make sure that people think about that. When you're thinking about a discovery project, maximizing the range of samples that you have available maximizes your chance for success. So I like to make sure people can think about all the things that they could be collecting. There's a balance of cost, there's a balance of what can be done, but I think it's important, as I'll talk about discovery or targeted studies, to know if you can, what you want to ask, or if you're really trying to create a resource to go for breadth. The other thing I wanted to flag is peripheral blood mononuclear cells called PBMCs. there can be really very nice advantages to be able to collect these, and they can be easier than the collection of what we used to do, which was skin fibroblasts. So if you're familiar with induced pluripotent stem cells, again, a lot of interest around stem cell technology, looking for cells that have the ability to differentiate into a number of different subsequent cells. Historically, we did a lot of work collecting those samples from skin, but we can now collect a great starting source, PBMCs, from blood samples. And if you're collecting other samples, again, this might be something you haven't thought of, but want to make sure that if you're thinking about the opportunity to be able to do stem cell work or have collaborators that might, stem cells and iPSCs can be very valuable when you're thinking about potentially evaluating some drug treatments, wanting to be able to do modeling. That can be very powerful, and it's something that often we don't think about. So wanted to make sure that I highlighted the importance and value and that opportunity to collect that from a blood sample. You also heard quite a bit around cerebrospinal fluid. Again, that's been an incredibly powerful source of sample and biomarker assays. As you saw in the Alzheimer's field, a great deal of work started in CSF, but then there was a great deal of interest in then being able to pair that work with CSF to then also look at a blood-based sample and see if we can move from a sample that's more challenging to obtain in a broad population to something that we can more easily obtain again from a blood sample. So hopefully just sort of giving you an overview and a sense of what we could be thinking about around samples. So sometimes, and again, I for some of you, this may be old news. I find that when I talk with investigators in a variety of biorepositories, there's incredible variability in what people know about samples, how to collect them, pros and cons, and what can I do easily or not easily in a research setting, sitting in, for example, in a clinic. So wanted to flag sort of as you go across complexity of sample and what you can do with them. DNA often is one of the easiest samples to collect. If we're doing it from blood, from saliva, relatively easy. It's a sample that can be collected easily, doesn't need to be frozen, is very robust. Depending on what you want to do with plasma and serum samples, I'll talk a little bit about it when I show you some protocols. Sometimes speed of processing becomes very important. And so we'll spend a little bit of time talking about that. And I've just kind of given you a feel of tubes that people typically use. RNA, again, a very easy sample to collect. Doesn't need to have rapid processing if you collect it in a PAX-gene tube. And PBMCs can be a little bit more complex depending on what you want to do with that. So that could be a sample that, depending on the capability of your site, the ability of being able to process that, could be a little bit more challenging. So just sort of wanted to lay that out across that collection of samples. One of the things that I think is really important when we're talking about samples and thinking about people starting out in clinical research is best practices. One of the biggest challenges I've seen with researchers is they've collected a set of samples, but the way they collected them was not standard or wasn't aligned with best practices. And then what they're able to do with those samples and the conclusions they can draw are then limited because of the way those samples were collected. So I wanted to flag that there are a number of resources, depending on the area that you're in, that you can seek out. And I put one example right here about making sure, as you're starting to think about collecting of samples and what you can do with them, about learning about best practices or partnering with other individuals who are familiar with those. Again, the time you're taking to collect those samples, the time your patient, now a clinical research participant, takes to provide those samples, we want to make sure that they can be used effectively afterward. So there's best practices around the collection, processing, storing, retrieving of those samples. Again, hopefully that's something that you're looking for more information on. This, I'm certainly glad to provide. I know Jeff would, and I'm sure others in this program would be glad to provide. Just really want to flag that. Ethics is flagged there too, and I'm going to mention that later on. How you consent individuals is incredibly important for what you're able to do with those samples. And often people don't think about what they might want to do, and they're hampered later on because their informed consent does not include the ability to do some of the studies or some of the partnerships that they may want to do. So I think it's important to try to think proactively and create, if appropriate, the greatest breadth of opportunity for how you'll be able to use those samples going forward. And I'll talk about that a little bit later on as well. So I wanted to flag something that years ago, I think perhaps people didn't really think about, but proved to be a real challenge. And just as Jeff tried to highlight things that we've learned, sometimes the hard way. I wanted to flag the concept of tubes matter and why best practices exist. So early on, people were using a variety of tubes when they were collecting samples for cerebrospinal fluid. So they would aliquot them. So after they did their LP, they would then place various volumes of CSF into a variety of tubes. And people weren't necessarily thinking, nor did they have necessarily the experience to worry about what kind of tube that they were using. But as the data began to grow around A-beta assays, and again, I do a lot of work in the Alzheimer's disease space, it became clear that the composition of the tubes was incredibly important for what we learned from the assays that were performed later on from those samples. And in fact, what was occurring was A-beta was sticking to the walls of the tubes. And so your ability to obtain reproducible results off of samples, the same subject collected at the same time in different tubes would obtain different assay results. It also became clear that as you transferred the sample, so if you moved from a larger tube into smaller tubes, that you also could have some loss of protein on the walls of those tubes. So what happened is it became very clear that recommendations were needed and best practices were needed. And so very quickly, in the Alzheimer's disease field, it was clear that low binding tubes, typically polypropylene, were essential for the collection of CSF if you wanted to be able to do A-beta assays later on. I wanted to particularly flag this example to highlight the importance of looking at best practices, and that best practices can differ depending on the area of study you're in. And so it's important to understand general best practices and also within your field, and to ensure that whether there are some differences or more specific limitations as you move into a particular disease area. So I'm hoping that that provides a little bit of context and gives you a concrete example, and I'm glad to go into more detail around that. So once it was realized, and again, along that idea of what do we do and how do we develop best practices? Within the Alzheimer's field, you've seen from both Jeff and hopefully you've seen from other speakers, the importance of, we do a lot of work often in a certain sample type. So in the case of CSF in the Alzheimer's world, and then the goal of trying to move to blood-based assay development. So there are groups, because of what we've learned in the Alzheimer's field, who have particularly worked to review and compare pre-analytical variables. So again, this leads to, which I'll show you in the next slide, the development of standard operating procedures. What is the best way to collect and handle samples? So the kinds of things these groups look at are sample factors, how you collect the sample, how you process the sample, that's what preparation means, and then how you store the sample. And I flagged in this particular case, this paper, but I want to show you what best practices can lead you to in terms of protocols. So on this slide, hopefully it's a lot of information, but I've got, so not necessarily need you to see all the details here, but I want to make sure you have the reference here. What I want to give you when you think about best practices, and again, the importance of adhering to these and the challenges, as I'll highlight, if they aren't adhered to, is for the collection here, for example, of plasma, what we have here is recommendations around what kinds of tubes we want to use, okay? And you see other sample types may have different biomarker levels, and you might need to use different kind of tubes. Again, there's always that caveat. Do you know exactly what you want to study? How specific do we need to be? And again, protocols that ask if you're going to be handling that sample and if you're going to process it within 3 hours, we can do this in a centrifuge that's room temperature or cold. There's specific guidelines around centrifugation, specific guidelines again around polypropylene tubes, what kind of volumes we want to do, and then how you want to store those samples. Also information about freeze thaws. So in the case of Alzheimer's disease and in many other areas, people have done extensive work to understand what parameters can I change? What parameters do I have to keep constant in order to obtain samples that will give reliable results consistently over time for a particular assay? And so I want to make sure that you're aware of this. And as you go and continue your careers, that you're attuned to asking in the area that I'm in, what are the best practices? Where can I sustain variability without impacting what I can do later on? Just really want to highlight the resources that are important to know about. So I also want to flag, I think many people come to me and say, like, what should I collect? And I think there's two different scenarios that I think are important to maybe talk through. Are there specific planned analyses that you want to do? So are you going into this with a collaboration? This is specifically what we want to do. Then you want to stick to the collection that's going to be needed to answer those specific questions. But I would note that there's often cases, and I mean, probably the majority of people I talk to are banking for the future. They know that they want to study this research area, but they don't necessarily have specific questions. They really want to create a resource, a bank, if you want to call it that they can then partner with others to be able to use those, or perhaps advance their own research. But it's more unspecified use. They're not quite sure what they're going to use it for. So I wanted to have a little bit of discussion around what I'd recommend in that setting. I think a broader range of samples, if you can afford it, if you can do that, can be really valuable in that situation. It creates more value, allows you to compare. Like if you're thinking about developing a biomarker assay, will it be better in plasma or serum? You can optimize it and determine which gives you the best assay properties. Again, important to collect using uniform protocols that are going to give you the best opportunity to be able to ask a range of questions. So again, thinking about best practices. And this is where I wanted to pair it and just remind you again about the importance of the consent that you use. And let me say what I mean by that. The first is, how specific are you about what you want to use those samples for? So is it just for this particular disease? The more specific you are about exactly what scope of question you can ask, the more limited you are. And often research participants, if you are comfortable and have a discussion around the importance of having a broad range of questions and comparisons that you want to make, that can be important to have that generality in the consent. The other area I'd highlight that can often limit individuals is whether or not they're limited to academic collaborations only. Are they limited to your own institution only? What about collaborations with non-academic or industry partners? If you use very limiting language in your consent, you're tied to that long term. And if you want to be able to collaborate with someone at their institution and you haven't created some flexibility around that, that can limit what you're going to be able to do. And again, and the other possibility is there could be industry collaborations that could be really beneficial for the use of those samples and for you. If you haven't consented those individuals for that, again, that could limit what you're able to do. And so again, I think it's important to think broadly and recognize that you have limitations that you may be setting upon yourself that may hamper your ability to ask questions in the future. So what I wanted to do is pause here. It's 12:50. And so I have another part, but I don't need to do with that. That's around genetics. And so I really wanted to take a pause here because I think my main importance was to sort of be able to follow up with Jeff and talk about samples. And let me just leave a moment there to ask questions around that if I can. So I'll stop sharing. I'll look to see if there are any questions around that. Oh, okay. So there are some questions. That's great. Okay, so there's a question around what are the pre-analytical challenges regarding saliva and tears in terms of collection and storage? Also, are there any downsides for the use of these biosources for biomarker discovery? Oh, that's a great question. Okay, so let me start with that. Let me start with saliva first, if I could. So how you collect saliva in the same way can have the same limitations. So there are saliva kits that are commercially available that are specifically designed, for example, for DNA extraction or for RNA isolation, okay? And it's important that you know which one you're using because that's going to determine what you're going to obtain from those samples. You can't use an RNA kit for saliva to then go do DNA extractions and go do genotyping and vice versa. So it's important to know which one you're using. The next thing I'd say is, so I work in the Parkinson's disease space, and there's been interest in various studies to do what's called passive drool in order to be able to look at biomarkers around, again, just like you were describing in saliva. You cannot collect saliva using one of these DNA or RNA kits if you want to be able to try to do biomarker assays out of a passive drool kind of collection. Again, it's a different kind of collection. The individual is just sort of holding the tube, and it takes quite a bit of time for that saliva to literally be able to go in that tube. So incredibly important to know what it is you're going to be doing because there are commercially available kits that when you purchase them, you know exactly what you're going down in that thread. The other thing I'd say is often saliva is a more limited volume. So your ability to do a variety of assays is going to be more limited as compared to if you collected blood. If blood can also be used for the assays you're thinking about. Easy to collect multiple 10 ml tubes. In saliva, in a lot of these kits, it's a much smaller volume. Now for tears, I have less experience. I have tears from one study in the bank that I have. Again, it's a pretty limited resource. And so that to me is one of the primary downsides that I think is really a challenge there. And I'm hoping I answered that question thoroughly. I'm glad to, if you want to ask me any questions to clarify more. There's also a question about references. Let me just slide that down. References around informed consent. So I will say I have got certainly the biorepositories that I have, we have some recommended language around informed consents. I'm also glad, I'm looking at Robin, and I hate to put you on the spot, Robin, but you might also want to point us to, from your past life, around NIH resources that you think are particularly good around recommendations for consent language. And if you have something that you know right off the top of your head, if you want to pop that in there. Otherwise, I'm certainly glad to provide links to the repositories that I run that have some nice broad consent language, and I'm glad to provide that as well. It's incredibly important. I'm glad to see the question because I think that then resonated with understanding that that's really a key factor in designing a study.
Moving on to Daniel, you have your hand raised, so I allowed you to talk.
Thank you. This is just a comment, Dr. Foroud. And to
Hello, Daniel.
Yes. So by 2027, Dr. Foroud will have 1,850 blood samples from a study I do. My only comment is, and I'm from Chicago, I would contact Dr. Foroud early, and I would contact her often if you really want help. Not only is she very good at this, and you can tell from her talk, the staff at BioSend are quite exceptional.
Thank you, Daniel. And I was not doing a shameless plug for myself, but that's also, if you're unsure and if you have NIH funded research, both NIA and NINDS, I think are eager to have samples that can also be shared with other investigators because it's incredibly important to be able to try to maximize the availability of samples for the breadth of research. Any other questions?
Looks like there's one other question in the chat.
Let me go
Oh,
look for it.
maybe you answered it already.
I think I did.
Yeah, this was absolutely great. Tatiana, thank you so much. This is Lori, sorry, I guess you can tell. I just realized my name's at the top while I'm talking, but I think this has been really helpful. You and Jeff combined, it's been one of our best biomarker talks that we've had in the course of the decade that we've been running this course, and just truly appreciate your willingness to share all your knowledge with us.
So I'm glad to do it. Can I ask him my last? I think there's one more question. Can I do it? Okay, is that all right? Corinne? I think you're unmuted. She's got her hand raised.
Yes, I allowed you to talk, Corinne.
We might be having trouble. Corinne, do you want to pop your question in the chat? I can look there too if that's helpful. Oh, it looks like there was a question for Jeff on assay validation. I think we're going to have to, if we can get the question, if someone wants to type it in, we can certainly get it to him. I think he's gotten off though. Maybe Corinne doesn't have a question. I think we might be done. Maybe I should have let you stop where you were, Laura.
I wasn't trying to stop you. I mean, if we still
No. It was a nice place.
I'll just echo Lori. Thank you both for superb talks today, and I will send Courtney something for distribution on ICFs.
Thanks so much, Robin. I appreciate it. All right. Have a great holiday. I admire all of you for coming on just before a long weekend. Thanks so much.
I'll send out those links. Dr. Conwit send when I send out the recording. And thank you all so much for attending. If you can please take the evaluation, I have posted the link to the Qualtrics in the chat. And if you have any more questions, you can send them to me and I can send them out to the presenters. That question for Dr. Dage, I'll send to him in an email. So thank you so much. You guys have a great day.